Hello everyone. Today, I am delighted to present our research on gut microbial enrichment and dysfunctions upon microplastic exposure. Microplastics are widely present in the environment and human. Microplastic exposure induces selective enrichment and dysfunctions of gut microbiota. Besides, gut microbiota mediates human health. However, molecular mechanisms underlying microplastic-induced gut microbial dysbiosis and dysfunction remain unclear, such as the impact of metabolic factors on gut microbiota-host interactions. Highlight 1. Microplastic-induced gut microbiota enrichment is dominated by bacteria within Eubacteriales. Highlight 2. The polyamine synthesis pathway is activated to maintain glutathione-spermidine homeostasis, while pathways related to energy and reactive oxygen species production are decreased. 3. Aromatic amino acid neurotransmitter pathways are disturbed. Highlight 4. Increases in enterolactone synthesis and cholesterol-derived hormone synthesis occur. Highlight 5. Bacteria within Eubacteriales contributed most to metabolic disturbances. Microplastic exposure induced gut microbial enrichment from the phylum to species level. 11 bacteria within Eubacteriales were increased, while bacteria within bacilli or Bacteroidota were decreased, indicating that microplastic-induced microbial enrichment was dominated by Eubacteriales. Besides, chain bacteria, archaea, and eukaryotes were associated with changes in viruses. Moreover, colitis occurred in microplastic-exposed rats. Metagenomics indicated that 269 genes related to metabolic pathways changed significantly, demonstrating systemic metabolic disorders in gut microbiota under microplastic exposure. Metabolomics showed that 27 metabolic pathways, mainly related to redox homeostasis, neural, and hormone functions, changed significantly under microplastic exposure, with big increases in several metabolites related to neural and/or hormonal functions. Therefore, we further explored changes in relevant pathways. Microplastic exposure induced changes in gut microbial glutathione and related amino acid metabolism. Increases in SPEA, SPE, GSP, and GST indicated the activation of polyamine synthesis to maintain glutathione-spermidine homeostasis. Besides, pathways related to energy ROS production were systematically decreased under microplastic exposure, such as the tricarboxylic acid cycle, oxidative phosphorylation, and degradation of fatty acids and aromatic compounds. The tryptophan-kynurenine and tryptophan-indole pathway were decreased, while the tryptophan-serotonin pathway was increased in gut microbiota under microplastic exposure. Besides, the tyrosine-thyroxine pathway, dopamine degradation, and phenylalanine-phenylacetate pathway were activated in gut microbiota. Nervous function-related pathways were altered. Moreover, enterolactone synthesis and cholesterol-derived hormone synthesis were increased. 80.29% of firmicutes contributions were increased, while 97.25% of Bacteroidetes contributions were decreased under microplastic exposure. Key species with increased contributions were mainly Oscillospiraceae bacterium, Clostridioides bacterium, and Clostridia bacterium, indicating key contributions of firmicutes to metabolic functions under microplastic exposure, especially Eubacteria. In summary, microplastic-induced gut microbial enrichment was dominated by Eubacteria, and correlated with the virome and colitis. The polyamine synthesis pathway was activated to maintain glutathione-spermidine homeostasis, while pathways involved in energy and reactive oxygen species production were decreased. Additionally, aromatic amino acid neurotransmitter pathways were disturbed. Besides, enterolactone synthesis and cholesterol-derived hormone synthesis were increased. Moreover, Eubacteria contributed most to metabolic disorders.